Everyone, welcome. This year I've had the pleasure and privilege of working with Dr. Eliza here. Our project was Pathogenesis of Corticotroph tumor development, and also want to thank Dr. Coates and Dr. Cheeseman as well for being involved in the project and kind of acting as my co-mentors. So before we get into the talk, there was also a couple of writing projects that we had this year, just to highlight. Earlier this year, we published a review article, Prolactinoma through the Female Life Cycle. That was in the Endocrine Journal. And more recently, in the June edition of Endocrinology and Metabolism Clinics in North America, it was an issue dedicated to Cushing's syndrome. And we were fortunate to publish this article in Mortality in Patients with Cushing's syndrome. So let's get into the topic of the project. So in terms of the overview of our project, so generally speaking, the genetics of corticotroph, when we say corticotroph, we're referring to ACTH positive tumors. The tumorigenesis is largely unknown. And even for that which is known, limited, the clinical significance of the genetics is not entirely clear. And so, as such, the options and therapeutic and treatment options that are tumor-directed are limited at this time. So our aims or our objectives, if you will, are to investigate the genetic drivers in the pathogenesis of corticotroph tumors, and possibly even discovering some novel mutations that have yet to be described along the way. We hope to correlate the genetic findings with the clinical picture. Correlating the genetics with the clinics, if you will. And then, ideally, the kind of the end goal of all of this is to identify new targets for tumor-directed therapy. So, better understanding of the genetics for better patient care. That's kind of what we're aiming for here. So just a brief overview. In terms of Cushing's syndrome, we really can break it down into being ACTH dependent and independent. ACTH dependent, which is what we'll be focusing on, it comprises about 85% or so of Cushing's syndrome. The other 15% or so being ACTH dependent, independent meaning coming from the adrenals and etiology. And out of this 85%, again, another 85% or so is originating from the pituitary, what we call Cushing's disease. And then the other 10 to 15% or so is coming from ectopic sources. So we're going to be focusing our talk on this. So the overall incidence is about 10 to 60% within pituitary tumors for being ACTH positive, depending on, this is what Williams' textbook quotes. And the incidence varies with gender as well as age. So in the adult population, there is a female 3-to-1 predominance, so about 75-25%. Whereas, interestingly, in children, it is an equal distribution, 1-to-1. And not all ACTH-secreting tumors cause this classical Cushing disease phenotype, which we see here. So, for example, we have silent ACTH tumors. These are tumors with few or really no biochemical or clinical features of hypercortisolism. This diagnosis is pretty much almost always made post-operatively based on the histological staining positive for ACTH. And these are generally larger, more aggressive tumors with higher incidence of recurrence, often needing radiation therapy. And we don't have any specific medical treatment available at this time for these tumors. Crooks cell adenoma is a rare, really incompletely understood tumor. It's defined based on the cytokeratin within the adenoma cells that is a result of hypercortisolemia. And if 50% of the adenoma cells demonstrate these changes, we refer to that as a Crooks cell adenoma, not to be confused with Crooks-Hyland change. And these are generally associated with functioning, but could also be associated with non-functioning tumors. And likewise, these are also very invasive and often aggressive tumors as well. And lastly, we're left with ACTH carcinoma. And And basically, ACTH carcinoma are very aggressive, invasive tumors that are really defined by metastasis. That's how we really define it. Now, in terms of the genetics of corticotroph tumors, starting off with germlines. So really, there are only a few familial or germline disorders associated with Cushing's. The ones that have been described out there, generally speaking, have been predominantly case reports and maybe case series at best. So things like MEN1, MEN2, MEN4 has like one case, for example, only a few cases associated with pituitary, only one of which is Cushing's. Familial isolated pituitary adenoma, and Carney's complex, associated with atrial lymphomas, some pituitary adenomas, often adrenal nodular disease, and pretty much for all of these, Cushing's is rare amongst all of these. And even when pituitary tumors are prevalent, it's generally speaking prolactinomas and growth hormone secreting adenomas. So really not getting much from these. Therefore, the majority of our mutations, therefore, are sporadic, also known as somatic mutations, meaning that they're arising from a clonal expansion of a single cell. And historically, it's been difficult in finding a recurrent causative sporadic mutation until recently, when in 2015, really a couple of papers kind of came out and changed our understanding and just really opened a new field in terms of Cushing's. So this paper by Reinke from Germany was the first paper to identify this USP8 mutation as contributing to Cushing's. And this was published in Nature Genetics in 2015. Around the same time, almost concomitantly, there was a paper by Ma and his group in China also showing that there is a pretty significant prevalence of USP8 mutations within Cushing's disease. And there's been a few other papers coming out since, and the most recent one is this one by Ballman, also from Germany. This one, I believe, was from Düsseldorf, and this was just from a few months ago, also showing their findings and their experience with USP8 mutations. So the USP8, it codes for an enzyme with deubiquitinase activity. And what that means is basically the end goal of it has to do with EGFR. It protects really EGFR from degradation, essentially. So more EGFR, more signaling, etc. And so when you have increased EGFR signaling with this USP8 gain of function mutation, it leads to increased POMC transcription and subsequently ACTH production among the corticotroph cells. Now, clinically, not a whole lot is known about these, but what is known and what this few studies out there have shown is that these are typically USP8 positive tumors are typically associated with smaller tumors, less extracellular extension. Females predominant even more so than they do in the general population with USP8 mutations. These are generally younger. And as I mentioned, because of the way it works in terms of ACTH production, these are generally more functional tumors. And so far in the literature, the studies have shown somewhere between prevalence somewhere between 21, as low as 21, to as high as 62% of ACTH tumors containing this USP8 mutation. And just a really quick image here, basically, instead of the EGFR being ubiquinated, essentially tagged, if you will, for lysosomal degradation, the USP8 is deubiquitinating these when it's overactive, and subsequently you get increased segue down this pathway going to ERK, NUR, and then subsequently POMC and ACTH. That's kind of what's coming out of it. So now the question is really, it's nice to hear about all of these genetics, but so what really? What's the clinical implications here? How is this going to affect our patients? And I have two really good examples of how we can translate this knowledge into something more practical and clinically useful. So this is a clinical trial currently undergoing in China, actually, and they're using gefitinib, using the knowledge of the effect of USP8 mutations on EGFR. Gefitinib is an EGFR inhibitor, currently FDA approved for non-small cell lung cancer with EGFR mutations. And so they're using this knowledge, targeting this, using gefitinib in patients with USP8 positive Cushing's disease. So we're eager to see what the results of this show. But one example of how we can utilize the genetics for kind of therapeutic purposes. Another example using this is this paper from Hayashi from Japan. This was from 2016. They focused specifically on the USP8 tumors. And they showed, interestingly, that there was increased expression of the type 5 somatostatin receptor among USP8 positive. So, does this mean that these patients are more responsive to an agent such as pasritamig? It remains to be shown, but it's interesting in theory, at least. Furthermore, they also showed increased expression of NGMT. NGMT essentially mitigates or antagonizes the effect of temozolomide. Temozolomide is used in various issues, most commonly in glioblastoma, but it's also used in very aggressive pituitary tumors. We see that as well. So two kind of interesting things that need to be studied more in detail clinically, but we can kind of see the thought process that can arise from knowledge of these tumors. So the question remains, though, if it's still a significant amount, 21 to 62%, but still a significant amount that is not being accounted for. So what are the drivers of USP8 negative corticotroph tumors? So the somatic mutations that we know, obviously, we know USP8. What else? T53 is something that's been described. We're going to talk more about that, but only 3 cases have been described in corticotroph tumors. The loss of function mutation T53. The GNAS gain of function mutation associated with McCune-Albright, where you see fibrous dysplasia and precocious puberty and cafe au lait spots, sometimes associated with pituitary tumors and hyperthyroidism as well. Only 3 cases have been described. Furthermore, this IN3CR1, it encodes the corticoid receptor. 2 cases so far have been described in corticotroph tumors, and this is a loss of function mutation. So coming back to our study, what have we been doing in regards to all this? So in terms of our study patients, we are enrolling adult patients, or what we have been doing is enrolling adult patients with pituitary adenomas confirmed with ACTH pathology. These are pretty much almost all have been operated on by Dr. Post, with maybe 1 or 2 exceptions. And pretty much everyone has received their care at Mount Sinai, again, maybe with 1 or 2 exceptions. No really other exclusion based on comorbidities, and our target recruitment has been about 100 patients. So the study design first was to develop a DNA mutation panel, and this was basically, the intention of this was to screen all ACTH tumors that we had in our study. And this was a custom ion ampli-seq targeted panel in collaboration with our genomics department here at Sinai. And this was basically handpicked by the cell biology, genomics, and our team, 53 mutations that have been implicated in involvement in the HPA axis, cell cycle regulation, and extrapolated from other cancers out there. And some examples you can see, for example, the EGFR and the USP8, but also some things like retinoblastoma 1, SSRT5, POMC, IMIN1, etc. And furthermore, the intention was to do whole exome sequencing really on as many patients as possible, in particular some of these more atypical or aggressive ACTH tumors. So here is just a screenshot of our gene mutation panel with the 52 or so mutations. And just a brief word on whole exome sequencing. Whole exome sequencing, unlike whole genome sequencing, is just focusing really on the exons. And these are the protein coding genes, accounting for about 1 to 3% of the genome. And most genetic disorders are found within, most mutations leading to genetic disorders are found within the exons. And so our goal with whole exome sequencing was, 1, to better characterize and potentially identify novel mutations associated with ACTH tumors, and furthermore looking at some other interesting genomic aspects such as CNV, copy number variation, which we'll talk briefly about as well. The only caveat with whole exome sequencing is that you do need a blood sample as a control to ensure that the mutation is tumor specific. So, getting the patients to come back, especially some had their surgeries done maybe over 10, 12, 15 years ago or so, was challenging in some cases. So in terms of what we've done, our study progress, which has been going on for a couple of years now, by Dr. Monell the year before me, Dr. Cheeseman the year prior to that, 78 patients with ACTH tumors have been consented and enrolled in our study. After subtracting, samples that are missing or inadequate DNA, what have you, we're left with 53 patients with adequate DNA samples for analysis. And among this cohort, we're seeing about 75% females and 25% males. So it's a beautiful, classic distribution of gender as we see in the control population. We have 8 silent ACTH adenomas. We have 3 Crooks cell adenomas in our study. 1 is also pending, and we have 3 ACTH carcinomas, which are quite rare as well. So our lucky number 53, we've so far have completed the mutation panel on 53 patients. This 53 gene mutation panel. And in terms of the whole exome sequencing, it's been done on 18 patients, 4 of which are still pending. And if you're wondering why is the discrepancy, it's because we had to get these patients coming in to come in for blood work, and a lot of these are, patients are coming from all over the tri-state area, Long Island, Connecticut, New Jersey, etc. So to talk briefly about one of the things that we focused on in our study, which really has been mentioned in a few studies, but we really honed in on this and really tried to look at USDA status compared to clinical and demographic characteristics in a really detailed and comprehensive way. So what we found in our study was that 29% of our cohort had this USDA positive mutation. And we see in comparison, it's on the lower side, but it's still within that range. And then using, in collaboration with the biostatistics department, which we'll see in the next figures, the Wilcoxon rank sum test was used for continuous variables such as age, and then for the categorical variables, the Fisher's exact test was used for some of the more kind of binary things. So we can see here, this is unpublished data, but we hope to publish it soon. This is our cohort, basically. We can see here the USPA negative population compared to the USPA positive, and here is the total. And we focused on a variety of things, including the age, gender, some of the tumor specifics, including whether it was atypical, whether this was, what was the status, looking at interesting diagnosis within corticotrophic tumors, which we'll talk about. The staining characteristics, PI67 and P53, no study has really delved into this, and also whether there was a history of apoplexy. So some things to focus on. The age at diagnosis, although it did not really reach the 0.05, it was marginally significant and definitely trending towards significance. We can see here clearly the USPA positive mutations were younger, and that applies to both the mean as well as the median when compared to USPA negative. Furthermore, we can see here that among USPA positive patients, 93% were female, and so that is, and only 1 of them were male. So that was seen there. Silent ACTHs. We had 8 silent ACTH tumors, and if you see here, none of them were USPA positive. This is consistent with the literature. There's not one case described in the world literature of a silent ACTH tumor that's USPA positive. And I guess it makes sense because this is a functional tumor, increasing EGFR, increasing POMC, and subsequent ACTH. So we do expect these tumors to be hyper-functioning in terms of hormonal production. Crooks adenoma, we had 3 cases, again, none of which were USPA positive. There have been a few cases described out there. One paper from Japan showed they had a pretty significant, like 15 patients with Crooks cell adenomas. But the highest prevalence has been described as like 10%, but it's probably maybe even a little bit less than that. But in our case, we didn't find any that were USP8 positive. And lastly, on this page, I want to focus on ACTH carcinoma. So out of our 3 patients, one of them had a USP8 positive mutation. This has never been described in the literature. First case, we do plan to publish this. So some other features that we looked at, the number of surgeries, the type of treatment they received, whether it was medical, radiation, the invasiveness of the tumor, presence of hypopituitarism, and we also looked at comorbidities associated. And again, this has not been described in relation to USP8 positive status. So invasiveness was statistically significant. Patients that were USP8 negative had a higher likelihood of having invasive tumors. Hypopituitarism, although not reached 0.05, it was close, 0.076, definitely trending towards significance. So USP8 negative patients had higher incidence of higher hypopituitarism. And then looking at our comorbidities, none of them reached statistical significance, but we basically see the trend for hypertension, diabetes, psychiatric disease, osteopenia, and cirrhosis, as well as, the prevalence was not so common for cardiovascular and DVT. But for all of these, we see that in our USP8 patients, USP8 negative patients, they had a higher tendency to have these comorbidities. So I think the kind of take home from all of this is that USP8 positive mutations, generally speaking, although we had, for example, the ACTH carcinoma, which kind of, is atypical certainly, but USP8 positive are, generally speaking, smaller, less aggressive, associated with younger females, whereas some of these more aggressive tumors with higher recurrence involving hypopituitarism and some of these comorbidities even possibly are associated with USP8 negative. And we hope that by increasing our N and our study number of patients, we can even show that some more of these factors are clinically significant or statistically significant. But we certainly can see the trends here. Now, in terms of the mutations, some of the notable results thus far is that 2 novel mutations have been discovered based on the whole exome sequencing. That's the RAS-T1 and the ACK1. We'll talk briefly about both of those. Furthermore, TP53, we'll talk about that. Only 3 cases have been described in literature, and we plan to add 3 more cases to this body of knowledge. And then some preliminary findings which the genomics team kind of gave us the disclaimer that it needs to be, confirmed and what have you. But 2 cases of NR3C1, 2 cases of GNAS, which we saw was associated with Albright, 1 case of MEN1, 1 case of KR1A associated with the Carney complex, and then 1 case each of KBOF1 and DICER1, both of which have been implicated in Cushing's disease. DICER1, there's never been a case of Cushing's from pituitary adenoma, but they're from like a pituitary blastoma, which is like an embryonal Cushing, very rare. But we're further kind of investigating all of these mutations. So in terms of RAS-T1, there was I think it was actually the first patient in this study, was a triplet, 3 sisters, one of which had Cushing's disease. So interesting family. And this patient was found to have USP8 positive as well as a novel mutation, RAS-T1. And this is thought to play a role in POMC expression and ACTH secretion. This was published by the group, including Dr. Cheeseman and Dr. Geer, Dr. Post. And this was from last year in terms of this novel mutation. ACTH1, there was another interesting family. It was a father and son, both of which had Cushing's disease. They were both USP8 negative, RAS-T1 negative, but they were found to have a mutation by the name of ACK1. And this has been implicated in a variety of malignancies, and it's a oncogenic driver. Pathways have been implicated in cell migration, proliferation, and survival, and possibly even EGFR action as well. And so our colleagues at SUNY Farmingdale are currently performing functional assays to confirm the pathophysiological significance of this within Cushing's. Just to talk a bit about some of these TP53 mutations, again, only 3 of these have been described in literature. These are 3 more. TP53 is interesting because it's been implicated in about 50% of tumors overall. It's the most common mutation out there. But in corticotroph tumors, it's very rare. Again, only 3 cases. So our 3 patients, basically, the take home from all of these 3 patients with P53, all of them were USPA negative. All of them had macroadenomas, so large tumors. One of them was a silent ACTH, the other 2 were functional. 2 of them had high Ki67 staining. All 3 stained positive for P53 in immunohistochemistry. These are 2 distinct things. So just because you stain positive on immunohistochemistry when we read the histopathological report, doesn't mean that they have this P53 mutation. But in this case, they all stain positive. They all required additional treatment other than surgery, and in this case, very aggressive, requiring 5 transtenoidal surgeries, as well as XRT in a couple. All of these were invasive tumors, and 2 of them were active and 1 in remission. Now, so again, another example of how this can be utilized, clinically, and in terms of helping our patients, patient care. So there's currently no targeted therapy against the P53 mutation, surprisingly, even though it's so prevalent. But this paper just from a few months ago highlighted that there are a number of agents currently being studied, 2 of which I highlighted here are actually in phase 1 and phase 2 clinical trials targeting the P53 mutation. So something to kind of be on the lookout for the future as well. And again, using the better understanding of the genomics for better patient care. The last thing which I'm going to focus on is something that is known as copy number variation, CNV. So this is defined as a DNA segment. Often they quote greater than 1 kilobase that's present at a variable copy number in comparison to a reference genome. And these are generally duplications or deletions of the sections of the genome. And this has been linked with a variety of diseases or so, especially in the past 10 years or so, 10, 12 years, namely various neuropsychiatric disease such as autism and even ADHD, for example, among others, Huntington disease, Fragile X syndrome, etc., and a variety of malignancies as well. Now, some have even stated that increased CNV can potentially correspond with increased disease severity and perhaps even earlier onset of symptoms. And even in certain malignancies such as myeloid neoplasm, such as AML, CNV is being used in certain cases as a prognostic marker. However, in terms of ACTH tumors, really the involvement or the implications is really little to nothing is really known about this. There was really literally only one paper from I think 2016 or 17 from the Brigham, along with Dr. Dunn. They looked at 42 pituitary macroadenomas. But the thing to note is that only 5 of these were ACTH tumors. And they found that 29% were what they call disrupted tumors, meaning significant copy number variation, which they described as greater than 6% of their genome. And out of these 5 ACTH tumors, they didn't really focus on this, but basically extracting it from the paper, 4 of these 5 were silent ACTH tumors. And out of these 4, 3 of them had significant CNV, meaning disrupted tumors. Now again, we don't know exactly what that means yet, but we decided to look into that in our patient population. And in our cohort, based on the whole exome sequencing, we saw that patients that had USPA positive had significant less number of CNV here, so less overall copy gain and copy loss. And to say it another way, USPA negative patients here had significant number more of CNV. So does this correspond to disease severity, etc.? A good question. And this is pretty much showing the same thing. These are your USPA positive patients with less loss and gain in the red and blue, and the USPA negative patients with a lot more color and intensity. So moving forward, what are we kind of hoping to accomplish with this project? So one, we want to confirm our preliminary findings, whether the genomics, and also functional interpretation of some of these findings. We want to continue with our patient recruitment to increase that N and hopefully achieve statistical significance with some of these variables that we do think are significant and are related. Looking into something like methylation, performing methylation array, which is a surrogate marker for gene expression on these, as well as RNA sequencing, looking at, for example, fusion mutations, and that has not been described at all. So in terms of our take-home, so the pathogenesis of corticotroph tumors is really still largely unknown. We know that about 21 to 62% of ACTH tumors have this USDA mutation. But otherwise, as we can see, everything else is, case reports and what have you. So there's still a lot to be discovered, particularly when it comes to USDA negative mutations, and then some of these more aggressive tumors such as silent, Brooks, and ACTH carcinomas. And we need more understanding regarding the clinical significance of these genomic and genetic findings in correspondence with its clinical significance. So the genetics and the clinics, how do they correspond? And identifying, what we hope to do with all of this is improving patient care and identifying potential targets for improved therapeutics. So to summarize everything, really we want better genetic understanding for better patient care. So this is my references, and I'll be happy to answer any questions that you have.
I think you may save it to the end. I just want to give Stephanie in the time for it. So it's okay. So poor Stephanie has laryngitis, so she'll be using a microphone to do her presentation right now. Everyone, I think, knows Stephanie. She's another one of our second-year fellows, and she's had the opportunity to work with Dr. Davies for this past year on her research project.
Hello. Good morning. Thank you very much for being here, and I'm very excited to present my project. My mentor is Dr. Davies. I'm going to jump right in with our hypothesis for this project. Can everybody hear me okay? Okay. The hypothesis for this project is thyroid antibody positive women have a deficient Treg response, T regulatory cell response to pregnancy compared to women who are antibody negative. About 1 in 5 to 10 pregnancies ends in a miscarriage, and about 5% of couples have recurrent miscarriages, which is defined as more than 2 miscarriages in a year. And the cause is often unclear. We know that chromosomal abnormalities play a leading role, but what we do know for sure is that there's an association with autoimmune disease. Potential mechanisms include subclinical hypothyroidism, the direct effect of thyroid antibodies, and immune failure. I'm going to touch upon these topics briefly. So with regards to subclinical hypothyroidism, in a national US cohort with 5,400 participants who had subclinical hypothyroidism, 60% of these participants were treated with levothyroxine. And what the study showed was that treatment was associated with a decreased risk for pregnancy loss, which is great. But there was also an increased risk for preterm delivery, gestational diabetes, and preeclampsia. And the benefit was also only observed among women who had a pre-treatment TSH between 4.1 and 10. With regards to the effect of thyroid autoantibodies, we know from women who have antiphospholipid syndrome that they are at high risk for recurrent spontaneous miscarriage, preeclampsia, placental insufficiency, and intrauterine growth restriction. The mechanism includes that the antibodies attack the decidua and the trophoblast, which leads to a local but also systemic inflammation with infiltration with neutrophilic cells, complement C3 deposition, which then leads to inflammation and pregnancy loss. Now to immune failure, which is our topic. This is a study, it's actually a very old slide, it's already, the figure is 25 years old, and but it's actually still the best that's out there. It was conducted by Dr. Davies and his team in the 90s, and shows that women who are positive for thyroid autoantibodies have doubling rates of spontaneous miscarriage. And so the team concluded that antibody positivity in pregnant women is an independent marker for an at-risk pregnancy. And this finding was confirmed several times over the years. Being positive for antibodies against the thyroid also increases your risk for preterm delivery, which has been shown several times here in a study from Belgium, but also in Pakistan and Italy, with women having more than doubling rates, actually in some cases, for preterm delivery. So how does the immune system come into play here? We have here the thymus, where the T regulatory cells mature. They're called so-called thymus T regulatory cells. However, if they're released into the bloodstream as naive T cells and then mature in the periphery, they're described as peripheral T regulatory cells. T regulatory cells were formerly known as T suppressor cells, and they only make up a very small fraction of the immune system, but they play a very important role in containing the effector cells of the immune system. They play a role in autoimmunity, in suppression of allergies, in self-tolerance, and of course in tolerance of the fetus, which is in part made up of paternal genes, and thus foreign to the mother. We're also going to talk about these T helper 17 cells, which are very important effector cells of the immune system. Not much is known about the difference between peripheral T regulatory cells and thymic T regulatory cells, but it is known that the P-Tregs recognize paternal antigens and suppress maternal effector cells, whereas the thymic T regulatory cells suppress autoimmune response. So what do we know about the normal physiology of T regulatory cells in pregnancy? This is a very important figure to understand. As I mentioned, the fraction of T regulatory cells is just very small, only about 1 to 3%, and this is showing a non-pregnant state with the status being very low, or the proportion of T regulatory cells being very low. As soon as a woman becomes pregnant, there's a response in T regulatory cells which start to increase and peak in the second trimester, then slowly start to level off and then normalize in the postnatal phase. This is important to allow the fetus to grow, given that it is foreign. So here are a couple of things that happen. The T regulatory cells expand. They also expand within the uterus. And it is the responsibility of the T regulatory cells to suppress T helper 17 cells and to keep them down. There's a certain ratio that needs to be maintained throughout pregnancy. And if this ratio is altered and there's a overpowering of the T helper 17 cells, then there's an association with spontaneous abortion, preeclampsia, and preterm birth. And this is just to show this figuratively once again. In cases of autoimmunity, the effect of T helper 17 cells is greater than that of the T regulatory cells, whereas in a healthy pregnancy, the T regulatory cells are able to suppress the T helper 17 cells. This study here looked at T regulatory cells and miscarriage. So looking at proportions of T regulatory cells in the peripheral blood, we have here the proportion of T regulatory cells in women who are not pregnant, in women who have just suffered a spontaneous abortion, and in women in their early state of pregnancy. And you'll see that women who have just had a spontaneous abortion have similar proportions of T regulatory cells as women who are not pregnant. In fact, in this study, actually a little bit lower. If you go to the decidua, part of the placenta, this finding is even more impressive. These are the proportions of T regulatory cells from women who have just had spontaneous abortion taken from the decidua. And these are the proportions of the T regulatory cells taken from women who underwent an elective abortion, who would have otherwise had a healthy pregnancy. And we see that the proportion of T regulatory cells is much, much higher in their placenta compared to women with decreased T regulatory cells. And this is what it looks like in this picture in the uterus. So we have the uterus here, we have the growing placenta, the cord, and the fetus. Here is an antigen-presenting cell presenting a paternal antigen to a T regulatory cell, which then gets activated, and through its activation, it suppresses these effector cells which surround the uterus. This is the case in a healthy pregnancy. However, if a woman does not have this important increase of T regulatory cells, then these effector cells are not suppressed and attack the fetus and lead to an abortion. So back to our hypothesis, antibody-positive women have a deficient T regulatory response to pregnancy compared to pregnant women who are antibody-negative. Our primary outcome is the T regulatory cell response in pregnant women. The secondary outcome are the pregnancy outcomes, which include which we divide into a successful pregnancy, miscarriage, or elective abortion. So far, we have been recruiting pregnant women from our high-risk OB clinic, mainly, who are autoantibody positive or negative, and we compare them with healthy non-pregnant controls. Our inclusion criteria are women who are aged 18 to 40 years at any time in their pregnancy. And exclusion criteria are any autoimmune disease such as type 1 diabetes, lupus, rheumatoid arthritis, multiple sclerosis. We obtain a peripheral venous blood sample. We evaluate their TSH and antibody status, and then isolate the T regulatory cells by magnetic bead sorting. We then stain the remaining cells with anti-CD4 and anti-CD5 antibodies, and stain them intracellularly with anti-FOXP3 antibodies, as anti-FOXP3 is the most characteristic marker of T regulatory cells, and then calculate the percentage of CD4, CD25, and FOXP3 cells. And this is what it looks like then in our FACS, when we see, here we have our T regulatory cells expressing high levels of CD25 and FOXP3. So far, we've recruited 55 participants into the study. We have 15 healthy controls, 40 pregnant women, of whom 17 are antibody positive and 23 are antibody negative. So far, with regards to the outcomes, we have had 16 healthy live births, 12 were full term, and we've had 4 early term deliveries and 1 miscarriage in this group of antibody positive women. The miscarriage happened at 10 weeks and 4 days, so relatively early in the pregnancy. Among the antibody negative women, we've had 23 healthy live births, 12 were full term, 5 were early term, and 6 were preterm. These were the conditions under which the babies were born preterm. These are some of our preliminary findings showing that women who are antibody positive have indeed higher levels of T regulatory cells compared to women who are antibody positive who have similar levels to women who are not pregnant. So in summary, pregnant women with thyroid autoantibodies are at an increased risk for spontaneous miscarriage. In a normal pregnancy, there's supposed to be a T regulatory cell response to this pregnancy. And pregnant women who are antibody positive appear not to be able to mount such a vigorous T regulatory cell response. the number of the T regulatory cells is decreased and the function is impaired. And we still need to learn more about the role of peripheral T regulatory cells. And we hope to do this with money which we have received through an ATA grant, which was awarded last week, which we're very excited about. And so hopefully more studies will emerge from this project. And I thanked Dr. Davies, who unfortunately is not here today, who has been an amazing mentor, and everyone else on the team who has helped me to be able to present this project today. Thank you very much.